Good morning. I'm Andrew Taylor, Vice President and the Immunology Microbiology Trustee. This morning, I'm pleased to review the history of the Cogan Award. The award was established in commemoration of the outstanding scientific accomplishments of David G. Cogan, MD, and recognizes a researcher who is 45 years of age or less at the time of nomination. The Cogan Award recipient will have made important and worthwhile contributions to research in ophthalmology or vision science that is directly related to disorders of human eye or visual system, and also exhibits a substantial promise for future contributions to eye and vision research. The 2024 Cogan Award is presented to Daniel R. Saban, PhD, Associate Professor of Ophthalmology and Immunology at Duke School of Medicine, Scientific Director of the Foster Center for Ocular Immunology, and the Faculty Network of the Duke Institute for Brain Science. Dr. Saban studies ocular immune cells, but with a unique emphasis on their non-immune properties. His research focuses on uncovering the significance of these immune cells in adult tissue physiology and disease resistance, and is leveraging his findings to develop therapeutics in eye diseases, including retinal degeneration. It is my pleasure to present to someone which I have watched grow since a grad student, this year's Cogan Award to you, Dr. Danny Saban. I am now greatly honored to introduce one of the most influential researchers in ocular immunology, Dr. Rachel Caspi, Chief of the Laboratory of Immunology, the National Eye Institute at the National Institute of Health, who will introduce Dr. Saban. Dr. Caspi.
It's like the third degree here. So, thank you all for coming and honoring Danny Saban. And Danny is today's ARVO award recipient, as you have heard, and he has been one of the most rapidly rising stars in ocular immunology research. He says he's already risen. And maybe he's right, because otherwise we wouldn't be all here. But I think that there's never a limit to rising. And like 50% of you here in the audience are probably going to be relieved that we still give awards to guys. So I have had the great honor of knowing Dr. Cogan personally during his last 8 years of his career at NEI, and he always placed a huge emphasis on personally mentoring and promoting young and upcoming investigators, and actively encouraged the cooperation between clinicians and scientists doing basic research. And you can see here in the picture, he is here with a group of his mentees, and that includes the late Bob Nussenblatt and Chi-Chao Chan, colleagues who have contributed so much to what we are doing today. So I know that he would have heartily approved of Danny as being a truly deserving person to get the award that bears his name. So Danny was born in New York to a family of immigrants who are sitting right here in the first row. His mother is from Poland, a daughter of Holocaust survivors, and his father hails from Lebanon. They met and married in Israel. So there is quite a remarkable history of diversity in itself here. Danny was the middle child in the family, and I'm convinced that this fact contributed to shaping his own career. Having to compete up and down the ladder in the sibling hierarchy, having to measure up to an older sibling, but also protect and be a big brother to a younger sibling, fostered his drive and ambition for achievement, and also honed his ability to work well with colleagues up and down the ladder, and to guide and mentor his own research group. Qualities that are so characteristic of Danny as I know him. Now, Danny's early mentors who shaped his interest in ocular biology and immunology were all towering figures in their own fields. After Wayne Streinein's passing at the beginning of Danny's PhD work, he was supposed to do his PhD work in Streinein's lab. He completed his degree under the guidance of Maria Grant and Amon Peck, and then he joined Reza Dana as a postdoc at the Scapins, and that was just about the time that I first met him. And it was already clear at that time that this young man was going places. While at Reza's lab, he developed novel approaches to study myeloid cell populations in the cornea, which then led to his subsequent long-term interest in myeloid cells and his seminal contributions on the role of the retinal microglia that he continues to pursue in his current position as an independent investigator at Duke University, that and a lot more that he will tell you about. So in his own lab, he utilizes such cutting-edge technologies as single-cell RNA sequencing, genetic lineage tracing, and his work has been instrumental in expanding our fundamental knowledge of ocular immunology and holds profound implications for therapeutic interventions in diseases like AMD and other retinal degenerative diseases. So not only have Danny's own contributions to eye research already been transformative, but he also mastered the so-called recipe for success that promises further outstanding contributions in his career. And this is a metal bottle. Must come from California. So he surrounds himself with collaborators who are recognized leaders in their own fields and whose expertise complements his own. And in that way, the product of these collaborations is more than the sum of its parts, which is exactly what we are all looking for. Now, these pictures show him with just a few of his many collaborators. I couldn't possibly put up all of them here. But looking at this happy crowd, you kind of wouldn't believe that these are serious scientists doing real serious science. But the contributions to science that they produce amongst themselves attest to it. So work hard, play hard, and produce groundbreaking research is the motto. So I'm not going to expand on the details of Danny's current work. He's here to do it himself. But before I give him the stage, I want to mention his service to the scientific community, to all of us, not only in his lab, but also in a more broader sense, in addition to the usual assortment of search committees and departmental committees and study sections and review articles, review activities. Danny has also served 3 years on the ARVO Immunology Microbiology Section Program Committee, and he is ready to take on additional roles to support ARVO and support all of us, should he be called upon to do that. He promised me. So finally, as I've heard Danny himself say, he would not be where he is today without the active encouragement and support of his own family, his wife, Melissa, and their son, Beckett, who are sitting right here in the first row together with Danny's parents, Iris and Jack, and sharing in his success and honor, as they well should, because they have contributed so much to it. So without further ado, I'm going to give the stage to Danny so that he can do what he does best and tell us about the science that brought him to this stage. Danny.
Water. Thank you, Rachel. That was wonderful. Wow. That was just incredible. It's amazing to be here today. It's amazing to be that this room is so full, and to be able to follow the previous speaker who already introduced the concept of macrophages. I'm just extremely pleased. So let me start talking a little bit about the science, okay? So I'm really going to focus on slightly different than the previous talk, which is the immune cells in the context of non-immune functions. And I'm calling them the unseen guardians of ocular health and disease, as you see in the title. And in the reason, and for this image as well, the reason that I'm showing this is that, when we study disease models, we study disease, thinking about disease in humans, we're often studying them in disease models rather than studying health. And so our emphasis, at least in the last 10 years, has been thinking about immune cells in the context of health, what they're doing in maintaining tissue structure and physiology, and how those basic elements, when lost, can lead to disease, and how, furthermore, we can intervene by replacing those elements, if possible, to bring it back to physiological states. And I just have one conflict of interest. I am a founder of a company, Piximune, which basically leverages this information that we've learned over the past decade to actually develop novel class of therapeutics to treat disease, which I'll touch on at the end. My timer isn't working, by the way, so I have no idea how slow or how fast I'm talking. There we go. Okay, so there's two faces of the immune cell physiology, as I've already alluded to. On the one hand, as most of you think about in the context of microbial defense, certainly we heard that about acanthamoeba before, wound healing, immune tolerance to sort of make sure we stave off autoimmunity. But on the other side is what we often don't think about, which is their production of growth factors, their role in tissue integrity, which I'll talk about today, and their roles in waste removal. And there are many more roles that are contributing to this system. So here's a great example of what we classically know, which is in the context of macrophages, which I'll be focusing on today. These are mononuclear phagocytes that live throughout our body, which we heard wonderfully about just prior to my lecture. So in the lung, you have these osteoclasts, which are macrophages involved in bone resorption and remodeling. In the alveolar space of the lung, obviously, we have surfactant clearance. They're constantly gobbling up surfactant for surfactant homeostasis. And of course, we know in the context of the central nervous system, we know about their role in synaptic pruning. These are all functional capacities that are critical in the success and function of the particular tissue. And baked into the cake in this context is that we now know from this landmark paper back in 2010 from Ginu and Murad and co. that microglia, your tissue-resident macrophages in the CNS, in the brain in this particular case, are not from the bone marrow. In fact, they're preceded, actually they're seeded from prenatal times, and they establish themselves prenatally and postnatally and actually maintain themselves in the in situ environment through being exceptionally long-lived and being able to self-renew. So there's no contribution of monocytes from the bone marrow into this population in the healthy tissue throughout the lifetime of the animal. And since then, a slew of high-impact papers were published essentially extending this idea to other tissues as well. So in this context, you see tissue-resident cells, tissue-resident macrophages. They're long-lived, extraordinarily long-lived. They're prenatal derived, and they can self-renew. And they seed the tissue prenatally and then maintain themselves without monocytic input for the remainder of the life of the animal in these tissues. So you have the microglia in the brain, the alveolar macrophages, the Langerhans cells, and the Kupffer cells. But that doesn't mean that the established model, the Van Furth system or model, was completely irrelevant, because we know that there are other tissue and niches within those organs that maintain monocyte-derived macrophages. These are from the bone marrow, They're short-lived. They circulate in the blood and then extravasate into tissue in the healthy sense. And these outpopulate the resident guys in these tissues and constantly turning over from the bone marrow. So I'm going to refer to these two populations as such. On the left-hand side, we have the long-lived tissue-resident macrophages, and on the right side are the short-lived macrophages, which I'll refer to as monocyte-derived macrophages. So what we wanted to do was essentially focus on these long-lived or tissue-resident macrophages with the hypothesis or prediction that if there was a population that was contributing to homeostasis in the tissue, it would likely be the long-lived macrophages, given their extensive residency in the tissue. And we looked at this question in the context of the cornea, Schlemm's canal, and the retina, which I'll be talking about today. So let's first start with our work in the cornea. So to really explain how we are able to study long versus short-lived or tissue-resident versus monocyte-derived cells, it's critical that I explain to you this mouse. I'm not going to really get into the genetics so deeply, but at least conceptually so you understand the model. So what happens with these mice is that all myeloid cells, macrophages, are YFP. They express YFP, so they fluoresce green by default. You then give them tamoxifen, which activates the TD tomato expression. So they're now not only YFP, but they're also TD tomato, so they're red and green. So what happens is that the short-lived or monocyte-derived cells actually die within 2 weeks or less. Okay, so you lose that population that was already made to be TD tomato positive through the tamoxifen. Whereas the tissue-resident, if you remember, our long-lived macrophages maintain the TD tomato, and so you can decipher these two macrophages in the tissue using this system. Not only that, but with the tamoxifen, it actually acts as a timestamp. So the moment you feed or give the animal tamoxifen, that is the time in which the TD tomato approximately gets activated. So you get a good sense of how old or the lifespan of the particular macrophage is. So now knowing that, I'll show you what we've done in the cornea. So here you have a intravital two-photon image of the cornea of a mouse, of this mouse. This is before the tamoxifen, and you can see the macrophages in YFP. And below, there's no TD tomato signal because they hadn't received tamoxifen yet. Once you give the tamoxifen, we now engage and activate the expression of TD tomato. This is actually 14 weeks post-tamoxifen administration, essentially telling us that these TD tomato positive macrophages are approximately 14 weeks old. That's essentially a long-lived macrophage. There's some nuances in this context which I won't get into, but suffice it to say that 14 weeks is indeed long-lived. So a very talented graduate student in my laboratory, Sajiro Littleton, wanted to study these cells and how they may potentially be playing a role in the cornea. So the first thing he did was do some single-cell RNA sequencing. He was able to quickly identify the corneal macrophages in the data set. He then took the data and plugged it into a gene ontology systems analysis, which annotates the expressed genes into pathways, and noted that some of the highest, most significant enriched pathways involved interactions with nerves, but also with glia. So this heavily suggested an interaction with the nerve components of the cornea. And this wasn't far-fetched because Paul McMenamin had done some classical studies showing that macrophages are indeed in and around the nerves in the cornea. So what Sajiro then did was, because of this data, he then found a mouse that expresses GFP, PLP-GFP on the Schwann cell, so these are the glia. He immunolabeled for TUJ1 on this flat mount as well, and then TUJ1 lets you see nerves. And then MRC1, which is a macrophage marker, and looked for places in the tissue where these three cell types interact. And he was able to find that quite uniquely at this basement membrane. So you see the Schwann cells, which are around the nerves, and then meet or drive them or help them or support them all the way until they get to the basement membrane, where they lose their Schwann cell interaction, and they now penetrate through the basement membrane to enter the epithelium. Right there where they're penetrating in many of these nerves, we see the macrophage. And that macrophage you can see throughout the cornea, or at least the central cornea in the image to the left at this what we're calling a tripartite cellular contact. And not only that, what Sugiro showed in the single cell data where the Schwann cells were making macrophage survival factors, and he then used a knockout of those survival factors and found that you had way fewer macrophages in this niche, suggesting not only are they in contact, but that the Schwann cells are actually nurturing and maintaining the tissue residence macrophages in that location. And this is a really beautiful image that he took in a live mouse. And again, not getting into the genetics, but specifically in this mouse, you can see all the macrophages, and incidentally, you can see about 10% of the nerves. Now, the blue is a rendering from the stroma. It's the second harmonic generation, so you can see where the stroma is. And all the red on top of that is actually in the epithelium, and you can see the 10% of those nerves, approximately 10% of those nerves, in the epithelial space. And then what Sugiro did was to render this capture so that you can separate the nerves and the macrophages, and you can see how closely, tightly interacting they are, or at least opposed, and specifically at that space. And so what Sugiro did was to actually deplete these macrophages to see what was going on, what their role may be playing in this context. And what he saw very, very robustly was that within 96 hours, many of these nerves, specifically in this epithelial area, were pretty much gone. But the interesting thing is, in this system, again, we were only seeing about approximately 10% of the nerves. So when we labeled for markers that told us where all the nerves are, it actually turned out that it was only a small percentage of the number of nerves that were degenerating in the absence of macrophages. So it suggested a select interaction between the macrophages. The other thing he noted was that when he did the depletion, there was no structural change or morphological change to the Schwann cells. So it suggested that the macrophages were specifically acting to support the nerves, but not through the Schwann cells. So thinking about this possible selectivity between macrophages and nerves, we started to do behavior analysis to see if we can sort of parse this out. And what Sugiro did was use aesthesiometry to look at mechanosensitivity in the context of depletion. And what he found is quickly, within 1 week, the mice lose their actual blink response to an aesthesiometer, in an aesthesiometry test. But the other interesting thing, and in working with Anna Matinia, who's done fantastic work with behavioral studies at the ocular surface, she tested other agonists to look at TRPV1, so this is heat, and TRPM8, cold, as well as hypoosmolarity and AITC, which are other stimulants. And there was no difference between the macrophage-depleted subset or mice versus the wild-type control. So what it suggested to us is that there was some selectivity. It wasn't that these macrophages were interacting or supporting every nerve. It was specifically these mechanosensory nerves. So this is the model that we're sort of working under, is that the tissue-resident macrophages are co-opting Schwann cells. As I told you, these Schwann cells are maintaining these macrophages to selectively maintain corneal mechanosensory terminals. So this is the idea. And we're doing continual studies to be able to try to prove our hypothesis, which we feel very strong about. So let me switch now to Schlemm's canal. This was work spearheaded by Katie Liu, a K08, who's started her own lab under the mentorship of myself and co-mentored with Dan Stamer, which has been a fantastic and a wonderful collaboration. So Katie was interested in the context of glaucoma, as our previous speaker spoke about connecting glaucoma with inflammation. So let me just bring a couple of points. We know that uveitis, for example, there's large sways in pressure, increases in pressure, linking inflammation and intraocular pressure. We know that immune modulating drugs can impact intraocular pressure. So for example, prostaglandin analogs like latanoprost are actually used at low doses to lower the pressure, whereas corticosteroids that are used topically increase pressure. So there's another link between the immune system and outflow homeostasis. We also know from clinical work that selective laser trabeculoplasty, SLT, had been proposed that monocytes by Alvarado et al. Had monocytes play an important role in actually this reducing effect. And then finally, GWAS studies had connected primary open glaucoma risk genes, had linked with GWAS studies macrophage pathways. So all of this suggested this possibility between the immune system and glaucoma, or at least outflow tract. So what Katie did was to immunolabel the outflow tract. You can see Schlemm's canal and the trabecular meshwork. She also looked at the distal vessels with these markers, CD31 showing you Schlemm's canal and the vessels, IBA1 telling you where the macrophages are, and you can see they're abundant throughout the primary outflow tract. And then we applied our fate mapping technique, as you remember, with the TD tomato mice, which enables us again to timestamp these macrophages. And the only place that she found these exceptionally long-lived tissue-resident macrophages, even out to 1 year, was in the trabecular meshwork. So it told us there was a unique enrichment of these long-lived macrophages in the trabecular meshwork, but not in Schlemm's canal or distal vessels, at least at the numbers that we see. So she went ahead and depleted these macrophages. So instead of expressing this TD tomato, she removed that gene and inputted a diphtheria toxin receptor gene, which allowed us to selectively ablate these tissue-resident macrophages while sparing the monocyte-derived cells. And what you can see is quite uniquely that in the absence, in the treatment with diphtheria toxin with the right genes, so we can ablate these tissue-resident macrophages, you can see a significant increase in intraocular pressure. And they went ahead and tested the outflow facility, which is the resistance in the proximal resistance of outflow. And the depletion actually reduced the facility, suggesting that we have a defect in primary outflow in the absence of these long-lived macrophages. And she's working hard now to figure out the mechanism, but what you can see on the left in a wild-type control, which wasn't depleted, is just beautiful distribution of the tissue cells, the trabecular meshworks, the vacuoles, and just a happy tissue. And in juxtaposition in the depleted context, you can see high compaction, you can see deposition right by the Schlemm's endothelial cells, and she actually quantified that and showed a significant increase. So it suggests that perhaps, this is again going to our working model, that these long-lived tissue-resident macrophages in the trabecular meshwork are contributing to tissue outflow or primary outflow through turnover of the ECM matrix. Again, yet another contribution of these cells in the context of tissue physiology. So in this last segment, I'm gonna pivot to the disease setting to address disease resilience. The idea is, now that I've given you some examples of how these cells can contribute to tissue physiology, I'm gonna push it one step further and suggest that these tissue physiological roles are actually maintained in the context of disease as well, and therefore contributing to disease resilience. So to understand this, let me just give you some background, because there are two potential populations of macrophages, at least two, in a diseased neurodegenerative context. So going back to the microglia, these are yolk sac derived, they self-renew, and they're long-lived, okay? And in health, they are compartmentalized from the blood circulation where the monocytes are coursing through. But in the context of an injury, the blood-brain barrier, the blood-retinal barrier is jeopardized. And they now pull through, they can come through the blood-retinal barrier Anyway, so they come in, they infiltrate, and it's very hard to tell the difference between these two populations. They are phenotypically, almost phenotypically indistinguishable, but yet their lineages are completely distinct and functionally are very, we now know are distinct as well. So we need to study these cells as two specific entities rather than one. So Emily O'Corn, who's a postdoc in my lab, who's actually here today, decided she wanted to use the fate mapping system to test the distribution of these cell types, the presence and distribution of these cell types. And so what you can see in the inner and outer plexiform layers where these microglia reside, you can see very nicely the TD tomato staining. You can measure these out to 1 year at 95% of all the YFP cells will be TD tomato or long-lived. It's remarkable. And she used the light damage model, which causes apoptosis of the outer set of the photoreceptors, essentially mimicking aspects of acute or retinal degenerative diseases such as IRDs. And in that context, what she found was, as we knew before, there was an increased number of myeloid cells, which you can see in the YFP segment. But remarkably, the only place where we found the TD tomato microglia was in the subretinal space. So there was some unique access and enrichment of microglia alone into the subretinal space. So she wanted to chase after that. And what she did, again, was to, instead of expressing the TD tomato, she expressed diphtheria toxin receptor. So you can specifically deplete microglia, but again, it's important to note that you're depleting microglia, but you're sparing the monocytes. And what you're looking at is a flat mount, the apical side of the RPE. You can see the phalloidin staining, which shows these beautiful hexagonal shapes of these cells in the normal. In the context of light damage, where you see lots of photoreceptor death, despite that, and with the presence of microglia attached to the apical side of the RPE looks remarkably healthy. But just juxtaposed to that, in the same model, but instead depleting microglia alone, you can see all heck breaks loose. There's massive atrophy, there's a strong level of dysmorphogenesis, essentially telling us or suggesting to us that microglia are playing a protective role in this setting. So this is a schematic. Microglia are moving to the subretinal space, attaching to the apical side of the RPE, and actually rendering protection rather than what was previously thought to be a more damaged type of response, just sort of with the assumption that these are contributing to neuroinflammation. So this was quite a shock to us, and I wasn't interested actually in studying the RPE, but Emily kept showing me this, and eventually I caved. So it's important, PIs, this is a lesson for the PIs, I guess, or the young PIs. Sometimes we have to listen to our students. But no, I mean, we obviously always have to listen to our students. Excuse me for saying that. So we published this in Immunity in 2019, and I think it was ignored for a little bit, but now it's sort of taking off, and there's a flurry of papers actually showing the same phenomenon in different contexts. There's work from Magali's lab showing in the aging retina of mouse. There's the RCS rat where it was showed. It was also showed in retinal detachment. In several of these different model systems, essentially showing this microglia population, which is protective. But still, the mechanism wasn't known. And so this is where Chen Yu stepped in, a postdoc in my lab now at Novartis, NIBR, who wanted to understand the mechanism of this protection a little bit better. We had already noted that the absence of these microglia was associated with the buildup of debris, a massive debris field. So we already knew that these microglia were playing a critical role in phagocytosis, or at least in the context of light degeneration. So what he did was, and the other aspect I should say about this is that when you look at the different mouse models of retinal degeneration, and whatever is available in human outer retinal degenerative diseases, whether it be a form of retinitis pigmentosa or age macular degeneration and geographic atrophy, for example, at least at the late stages, you see a remarkable similarity with the fact that you find macrophages accumulating in the subretinal space, similar to what I showed in the retina. So it suggested that there's some gene agnostics, movement of these microglia to the subretinal space, and suggested maybe there's some protective program that's conserved across these different settings. So that's what he set out to do. Using single-cell RNA-seq and leveraging these different models, the light damage model, the inherited retinal degenerative disease model, P23H knock-in, established by Chris Palchevsky, the sodium iodate model, which causes acute death to the RPE, and then in collaboration with Kathy Bose-Rickman, advanced aging 2-year-old mice, isolating, purifying the retinal cells, and running single-cell RNA sequencing, integrating the whole data set. He was able to find the subretinal microglia population, which we established by their gene modules in our immunity paper. And what was remarkable is that even in spite of these models being quite different, whether it's age-related and natural, whether it's acute from light degeneration or sodium iodate, we found a very similar or conserved signature, or transcriptional signature across all these models, which are highlighted in red. And one of the genes that was particularly upregulated that we were focusing on or became interested in L-GALS3, galectin-3. This is a galectin with roles both intracellularly and extracellularly. So what Chen did was to make the GAL3 flox mice so that we can conditionally knock out this gene from microglia specifically. And he found the same phenotype. That is, when you have a loss of galectin-3 in microglia alone, you see this massive RPE atrophy in the light damage model, as you can see on the bottom panel. And of course, we found this debris field, but you can see now with galectin-3, you can see the rhodopsin debris, which is from the photoreceptors, encapsulated in the microglia in the subretinal space, indicating that they've phagocytosed. Whereas in the absence of L-GALS3, you can see the rhodopsin is sort of diffuse and probably causing havoc to the outer retina. And he used this system, at least the global knockout system, and bred it onto the P23H mice as well, and then also looked at aging and found a very similar phenotype. That is, the degenerative aspects of what we saw were way more advanced in the absence of galectin-3. So with this remarkably conserved signature, we thought, okay, let's see, perhaps maybe there's some parallels in the context of humans in outer retinal degenerative diseases. And we were very fortunate to be able to collaborate with Nora Ladd and Alan Proya, both clinicians. Alan Proya, a board-certified pathologist who studies AMD, and got our hands on some postmortem tissues. Also very fortunate that Christine Curcio had her data set available online with, I believe it's Dwight Stambolian, and Rob Mullen's data set as well. So we can take our data and integrate them all together and look to see if there's potentially this conserved population of subretinal microglia in the context of human outer retinal degenerative disease as well. And so what Chen did was to use our mouse data to actually establish a 50-gene module so that we can mine the human data for a cluster that was similar to the cluster we saw in subretinal microglia, and he found one here referred to HMG2, that's human microglia cluster 2. And what was amazing is that this population had 87% conserved differentially expressed genes with what we saw in the mouse, strongly indicating that the functional relevance that we saw in mice was conserved in the context of human disease. And again, being fortunate to work with these incredible clinical colleagues who had access to tissues, we looked at about, I think it was N of 18, I have the bullet point coming up in a second, patients with geographic atrophy. You can see these CD68, presumably microglia in the subretinal space, exactly where you see the geographic atrophy. And we were actually able to use two other markers that were different, which were highly enriched in our microglia, TREM2 and L-GALS3, proving, or at least to the best of our ability, tying this in with the signature that we see in mice. So just to overview, this is a total of 39 donors from this data set. Again, this was integrated from Curcio's data set and Mullen's data set. Total of 40,000 or more myeloid cells from the retina and RPE choroid. Again, an 87% conserved differentially expressed gene signature between the mouse and the humans, and then the immunolabeling to validate our findings at the protein level with an N of 18 donors. So we feel very confident with our data set. So to now sort of summarize this last bit, what we found was that microglia have a unique access to the subretinal space. I'm not here saying that it's always and only microglia. Obviously, there are situations which have been shown by others where monocytes can be, but certainly in our hands, we see that there's certainly a major enrichment there. And these are not seemingly contributing to inflammation. They're able to clear debris and support the tissue in the relative absence of inflammation. And that's kind of a step away, a major step away from conventional thinking. So understanding the pathway, we are now able to develop a novel class of molecules that can actually switch on and augment this protective pathway. And we have a tool molecule that we've already published in mice, which is quite protective in the mouse context. And we're now working to humanize our approach so that we can potentially bring this to patients. So in total, I will summarize these amazing cells, the unseen guardians of ocular health and resilience. I showed you three unique settings from our laboratory. One is the conventional outflow, the second was the cornea, and the third was the outer retina in the degenerative context. And what you can see is a theme across all of three of these settings. That is, these macrophages are right there at a barrier tissue, a critical barrier tissue, to maintain proper function at that tissue. In the context of the outflow, it's an endothelial cell where the blood aqueous barrier is, enabling the Schlemm canal to support primary outflow. The ocular surface, it's right at the level of the tear film, below the level of the epithelium, supporting epithelial cells. We know that nerves are critical in supporting the epithelial cells. And I should have referenced Marianne Stepp's work in the original slide that I showed with the tripartite, because they had proposed a similar model in this context as well. And then finally, in the outer retina, between the retinal pigment epithelium, right below, and then, of course, outside is the choriocapillaris. So there's something unique about these cells in the context of barriers throughout the eye, at least in the three examples that we showed. So with that, I couldn't have done any of this without my fantastic team. I had mentioned Sajira Littleton and Katie Liu, Emily O'Corin, and Chen Yu, who are all critical in these particular pieces of data that I showed you today. Of course, my lab wouldn't be running day to day without Rose Matthew, who has been with me since the very beginning. Thank you, Rose. You've been incredible. And of course, I have to thank my incredible collaborators, some of which have already been named, such as Kathy Bose-Rickman, Maria Grant, who's also a collaborator as well, and Dan Stamer. All of these individuals that are listed here have been just incredibly supportive. And of course, the funding sources through the NIH, Bright Focus, whose money actually enabled us to enter the field of microglia, RPE CD8 Award and International Research Award, and then the P30, which supports our department and don't pay for some funding in our cornea stuff. I couldn't have done this without my nominees who took the time and supported me for this award. And You know, Rachel, thank you so much for leading this, and you've been a great supporter throughout my career. Andrew Dick, Deb Farrington, and Simon John, thank you. Mentors, Wayne Stryline just literally lit up my mind, cemented my love for ocular immunology, reading his stories about Acade, him and Jerry Niederkorn. That's really what made me tick, and still does. And thank you so much, Maria and Bruff Peck, for giving me the access and enabling me to grow and nurturing me, even though I was such a punk. And somehow you managed to carrot and stick me and keep me in the right direction, so thank you so much. And Ray Zidane, of course, who was my postdoctoral mentor, and Scott Cousins, who recruited me to Duke. And of course, you have close colleagues and friends along the way, some of which help you sort of parallel level, their colleagues, and others that are a little slightly more senior than you or much more senior than you, to give you advice and to slap you in the hand when you get out of line. And so I want to thank Sunil Chauhan, Sharmila Masley, Andrew Taylor, Victor Perez, my hermano grande, Eric Perlman, Jerry Niederkorn, Kathy Bose-Rickman, and Dan Stamer. I'm so appreciative. Thank you, Mom and Dad. Unconditional love. Mom, you taught me how to dream. You really did. Dad taught me sincerity, honesty, hard work, and the love of medicine and science. Thank you. Hilly, my sister, who's basically my second mom when Mom and Dad were working so hard during the early years, making sure I was happy, and my brother, who continues to just teach me things about life, inspiring me, and challenging me. There are no words. And thank you, Melissa. I couldn't do any of this without you. And the sacrifices that you and Beckett have to deal with, all the traveling, all the late nights, and the frustrations, but at the same time, all the fun and all the love that you bestow on our family is remarkable. And somehow I made it under the time, probably because I got a few extra minutes. Last thing I'd like to say is it's an honor to receive this award from Dr. Cogan's, on Dr. Cogan's name. I haven't met him, but Rachel tells me wonderful things, amazing things about his mentorship and his leadership abilities, his leadership and Victor also shared some stories with me, so I'm quite honored. Thank you for your attention and have a great rest of your trip. Thank you.